Good morning and hello. Hope you all had a good Easter holiday. Hope you were able to enjoy it with your family. As promised, today I will be going over some of the questions that you all sent me for the review session instead of starting a new module. So our next topic will be translation, but I will start that on Wednesday. Okay, for this review, unfortunately, I didn't hear from most of you. I only get questions from 3 students out of 140 students that we have in the class. So that was a very, very few students, of course, but those were great questions. And I am guessing that most of you probably have the same questions or similar questions. Okay, I have used the questions as such as you sent me, and I will be going over the answers to those. So the first question was if ethidium bromide can be so damaging to DNA and it can cause mutation. Remember, I mentioned that this is carcinogenic substance. Why it's commonly used in the labs. So that's the first part of the question. And second part of the question is, doesn't this pose a risk to the DNA that is being tested when we are running the DNA on the gel? Okay, so let's talk about the first part first. It's a molecule of choice. It's frequently used because it gives color under the UV light. It gives the fluorescence, and then you can see where the DNA is. So it's a very nice visible marker when you expose your gel under UV light. This nice fluorescence is because this is the that's what this molecule do, and it also stacks itself between the base pairs. So it does affect the DNA mobility a little bit, but it doesn't affect too much. And since we are using ethidium bromide with all DNA, so the mobility will be affected equally in all of that. So that's not really a problem. Why it's frequently used is because of this ability to detect DNA on the gel when exposed to UV light, because this ethidium bromide will give you that dye, that color of DNA that we see, the pinkish color on the gel. The second part of this question is, does this pose a risk to the DNA that's being tested? No, because that DNA that we are testing on the gel is already taken out of the cell. So we are not really mutating that DNA in any way. What you're really doing is just running it on the gel to see where it migrates. So mostly to see the size, mostly to see whether the DNA is present or not. So because the DNA on the cell is already extracted out of the cells, it's not really a risk for DNA to be mutated. However, there's a risk for the DNA inside our cells to get mutated because of ethidium bromide, because of the stacking between the bases. And that stacking, sometimes during replication, it can lead to indels, addition or deletion of a nucleotide, because it will increase the gap between 2 nucleotides. And that's why it's really important to use all sort of precautions that we need to in the lab while working on ethidium bromide. And those of you who are working in the labs, those of you are working with ethidium bromide, you probably have been told to use gloves all the time. Do not touch the gel or anything that contains ethidium bromide with bare hands, and proper disposal of those gels in ethidium bromide marked boxes, not in the regular trash can. There are some other dyes, that is ethidium bromide substitutes, that are non-carcinogenic, and a lot of labs have now switched to those ethidium bromide free dyes, and so in my lab we use those ethidium bromide free dyes as well. Okay, next question from the same student was, in eukaryotes, XPG cuts the damaged DNA. Is XPG considered an exonuclease? Yes, XPG is a nuclease. It has both 5' endo and exonuclease activities. So it can be an endonuclease or an exonuclease. Okay, so next question was that pyrimidine dimers can be fixed either by photoreactivation or by nucleotide excision repair, or NER. So how do prokaryotic cells know when to use which mechanism, so when to use photoreactivation and when to use nucleotide excision repair. So that's the first question. The second part of this question is it based on how extensive the damage is, since photoreactivation is a form of direct repair and NER is excision repair? So that's kind of, you're answering your own question here. So let's go over a little bit. Photoreactivation, it's a very common mechanism that prokaryotes use. In my lecture, I said that it's a prokaryotic specific. Eukaryotes don't use it. But then I was reading some more papers, and I realized, I learned this as well, that placental animals do not use photoreactivation. So mammals, humans including, will not use photoreactivation mechanism. But non-placental animals, for example, birds, this has been very well studied in chick embryos. And some plants also use photoreactivation mechanism. So just wanted to let you know here. Photoreactivation fixes is a direct repair, right? So it fixes the dimers caused usually by thiamine. And UV radiations hit. The UV radiations will make this, if there are two thymines together in the sequence, they will make them bind to make the dimer. Those dimers can be directly repaired by photoreactivation. NER usually is used for bulky damage, but NER does it removes the short stretch of the molecule, DNA molecule, including the healthy part of the DNA, and then it will repair the whole stretch. So it's usually about 30 nucleotides or so. One of the benefit of NER is, and why prokaryotes use these both mechanisms, with the thymine dimer, with the UV radiation, especially when there is a damage caused by UV radiation, it's photoreactivation that is the mechanism of choice. That's always preferred to use photoreactivation. When the damage is caused by other maybe other carcinogens, other environmental agents, that those changes, those corrections are usually repaired by NER, because NER in both prokaryotes and eukaryotes, it can recognize and remove a large number of DNA lesions that those might have been induced by a wide variety of environmental agents. These differ in chemical and structural composition, these environmental agents, but they can break the DNA molecule. NER is because it's called bulky damage repair mechanism. So when there is a lot of mutation going on, then NER is a mechanism of choice. These two I listed here, two different articles, and these are very, very nice article explain these repair mechanisms in great detail if you want to read more. So again, to answer your question specifically, damage caused by UV radiations is usually the prokaryotes will repair that by direct repair mechanism, so by photoreactivation. Damage caused by other molecules other than UV radiations is usually corrected by these base excision or nucleotide excision repairs. Okay, so these following questions are from a different student. And the first question here is from transcription. So let's see. It says the pre-initiation complex formation starts with TF2D binding the Teta box. So that's a TF2D bringing the TBP together. So because TBP is part of TF2D, it just binds, TF2D binds to TBP, brings it to Teta box. The Teta binding protein or TBP will bind to Teta box, and thus the pre-initiation complex starts from there. However, you also describe some genes, especially housekeeping and developmental genes, that lack that Teta box in their promoters. For genes like these that lack a Tata box, how does pre-initiation occur? This is a great question. So how does Tata-less promoters, that's what those are called, the promoters that do not have Tata box, how do the Tata-less promoters initiate or pre-initiate the transcription in eukaryotes. Okay, so let's talk about these promoters. So we talked about here the upstream of initiator is Tata box in promoters that have Tata box, right? The downstream of initiator is downstream promoter element, and I mentioned that these downstream promoter element and Tata box are either or. Usually the genes that have Tata box will not have downstream promoter element. Occasionally, in very few genes, they have both Tata box and DPE. But in majority of genes, if there is a downstream promoter element, there won't be a Tata box. If there is Tata box, there won't be a downstream promoter element. Okay. So this downstream promoter element is very important in Tata-less promoter genes. So the genes that don't have Tata box, they have this downstream promoter element, and this downstream promoter element binds to transcription factors in order to make the pre-initiator complex. So here, this diagram shows it pretty well. This is Tata box containing promoter, has this TBP binding to Tata box. Initiator will start initiation. These are those auxiliary factors 60 and 40. These TF2D binds to these TAFs as well. Here in the Tata-less DPE containing promoter, TBP still binds to the promoter region, to the upstream region, to some other sequence, not a Tata box. And here these auxiliary factors 60-40 bind to DPE, and this DPE then in a way it's binding of these auxiliary factors to DPE and binding of this Tata binding protein to promoter region here upstream. This together allows pre-initiation complex, together allows the initiation of transcription. These TAFs, these auxiliary factors, these 60-40, they play a direct role in basal transcription of Tata-less DPE containing genes. So these are required, the 60 and 40. This TAF60 tightly binds to DPE. TAF40 doesn't bind as tightly to DPE as 60, but these need to be both associated with DPE. Okay. There's another thing that is this paper here. If you want to read more about it, this is a really good paper. And this paper, which suggests here, you can see it here, that the Tata box is about 25 to 30 base pair upstream of initiation, where the transcription initiation happens, and downstream promoter element is also about 30 base pair, 28 to 34 base pair downstream of the initiation site. And this spacing is very strictly controlled. So there's a very strict requirement of spacing between the initiator and DPE motif. Because if there is an increase or decrease of 3 nucleotides in the distance between these two, that can reduce transcription quite a bit. 7 to 8 fold reduction can happen by moving this downstream promoter 3 nucleotides in front of the back. So, just to answer your question again, the promoters, the Tata-less promoters, require this downstream promoter element. Downstream promoter element is bound by these again the transcription factor 2D, and this transcription factor 2D brings these auxiliary factors 60 and 40. These 60 and 40 auxiliary factor need to bind to DPE. When these auxiliary factor bind to DPE, this Tata binding protein also binds to some sequence upstream of the initiator region. So that's not Tata box, and that's the reason I said in previous lecture that TBP is kind of a misnomer, because it's not really Tata box binding protein. It can bind to other sequences other than Tata box as well. So this TBP binds here, auxiliary factors bind here, and together these will initiate the transcription. Okay, so next question from the same student was about RNA processing. about alternative splicing in general, specifically. So the question is, you talk about how alternative splicing requires specialized splicing factors that bind to elements in exons, such as exonic splicing enhancer or exonic splicing inhibitors. Do some specialized splicing factors also bind to elements within introns for the regulation of alternative splicing? How important are introns in the regulation of alternative splicing, if at all? So the short answer to your question is yes. Yes, there are elements within introns that regulate alternative splicing. Answer to your second question is, how important are introns in regulation of alternative splicing? Quite a lot. They are very important. And you might remember one of the example we talked about is where the splice site, the branch site migrates in the intron, making the intron move, the splice site move, and that movement of that splice site will change the protein, the resulting protein. So let's talk about this mechanism in a little bit more detail since you asked this question. So thank you very much. Introns are equally important in the regulation of splicing. Okay. The effect of these splicing factors are position dependent, so it depends on where the splicing factor is. The splicing factor can serve as a splicing activator when it's bound to intronic enhance elements, or they can serve as a repressor when they're bound to its splicing element in the axon, or vice versa. It depends on where it's bound. So these splicing factors can be splicing activators, or these can be splicing repressors. Okay. The secondary structure of pre-mRNA transcript also plays a big role in regulating splicing, because remember, RNA is a single-stranded molecule, but it frequently folds over to itself to make secondary structures. Those secondary structures depends on how complementary the sequence is. So given the complementarity, it can fold over to make these secondary structures. The secondary structure can bring together splicing elements, because now this mRNA, it might be a long molecule, but once it's fold over to it onto itself, it can bring the splicing elements together. Or it can mask a sequence that would otherwise serve as a binding element for splicing factor. So that way it will be a splicing repressor. It will not let the splicing happen in that place. So together these elements make this something called splicing code that will govern how splicing occur under different cellular conditions. So whether this gene is going to be alternatively spliced or it's going to be not alternatively spliced can be governed by the splicing code, by these splicing element that regulate splicing. So these can be repressors, these can be enhancers. So here, let's talk in a little more detail. There are two major types of cis-acting RNA sequence elements present in pre-mRNAs, and they have their corresponding trans-acting splicing proteins. So you remember cis and trans. Cis is on the same DNA molecule, trans is on a different DNA molecule. So these trans-acting splicing proteins means these proteins can come from somewhere else and bind, right? So this is an example of splicing silencer, because this means silencing, this means block here. Right? So these are different elements. This is an intron, this is an exon. So I stands for intron. So this is an intron splicing silencer. This is an exon splicing silencer. So another intron splicing silencer. So when these are bound here, these intron silencing intron splicing silencers are bound here, these will not allow the silencing to happen. Okay, so these splicing silencers are sites to which splicing repressor proteins bind, and when these splicing repressor protein bind, they at splicing junctions, they can bind to either the intron splice. They can be intron splicing silencers binding into introns, or exon silencing splicing silencer that will bind to exon. So here, and these will then inhibit the splicing at that particular point. So this can inhibit splicing here. That means if this didn't get spliced out, that means this sequence will be different from its normal sequence. Okay, so here, this is example of splicing enhancers, because now these green arrows means splicing enhancers. So now the splicing enhancer proteins are bound. So this is intronic splicing enhancer, this is exonic splicing enhancer. So these proteins can bound in introns, this can be found in, they can bind to sequences in exon. So either way, intron or exon, they both are equally important. So these splicing enhancers are site to which splicing activator protein binds. These increase the probability that a nearby site will be used as a splice junction. Okay, so this will be spliced out. Most of these proteins they belong to SR protein family, just like we talked about these splicing proteins earlier. So these SR are serine arginine rich proteins. They bind to these splicing factors. These splicing factors can be inhibitors or enhancers. Okay, so your short answer of your question was yes, introns are equally important for alternative splicing as exons. Yes, there are introns have these specific sequences that the protein will bind to these sequences, and these can enhance splicing, or they can repress splicing events. Okay. So another student asked me to give some example problem sets that are related to the materials we have covered, whether it's a long response or multiple choice question. So I decided to give you a couple of multiple choice questions and one long answer question for practice. So I don't know how to give it for practice, so I will just go over these for now, and then you can think about it on your own later. So let's talk about transposable elements a little bit. Transposable elements cannot be made to target a locus, but they can still be used to knock out a target gene. How? How do you think transposable elements can be used to knock out a target gene? There are a couple of examples. There are a couple of answers here. So you have to, because it's a multiple choice question, you have to find the correct answer. So let's go over these answers. The first is, the element can insert at random sites, including the target gene, leading to knockout of the target gene. Okay. Next is, the element can carry a null mutant for the target gene and force homologous recombination. Okay. The target gene will eventually be knocked out if enough offspring are screened. None of the above transposons cannot be used to knock out expression of a specific gene. So let's go from D to A, make our way from bottom to up. None of the above transposons cannot be used to knock down expression of a specific gene. But that's incorrect, because we did talk about several examples where transposable elements have been used to knock down or knock out expression of specific genes, right? So transposable elements are actually the most commonly used, the most widely used way to knock out a gene to make transgenic animals or transgenic plants. So this is incorrect. So the target gene will eventually be knocked out if enough offspring are screened. Well, we can screen a lot of offspring, but if there is no knockout, if we don't use any way to knock out a gene, there won't be any knockout, right? We can keep screening, but if we haven't really done anything to that individual, there won't be any knockout. So that's incorrect. Element can carry a null mutant for the target gene and force homologous recombination. I don't know how the transposable element will force a homologous recombination. It can provide the sequence that we are using. It can insert that sequence, because that's what transposable elements do. Transposable elements, they jump from one place to another place in the genome, and once they jump there, they can copy themselves in another place if they're using that mechanism. But that's not really what happens when you're making a transgenic animal, right? So you're not really forcing homologous recombination, because that's not what transposable elements usually do. So the last of our answer is the element can insert at random site. That's what transposable elements do. They insert themselves at random sites, including the target gene. So, well, it's possible that if we inject it in a lot of organisms, their chances are, because of this their ability to randomly insert themselves, they will also insert themselves in some of the organisms. They will also insert themselves in the target gene. That will lead to knockout of that target gene. So this is actually the correct answer. Okay. Next question is targeted mutagenesis in mice. Is a kind of related question. Can be achieved through the activity of. Now, so targeted mutagenesis means we are trying to mutate a specific site in the mice. How can we achieve that? So homologous recombination using a disrupted copy of the target gene. Orphan transduction by retrotransposition, non-homologous recombination using a disrupted copy of the target gene, site-directed mutagenesis by in situ PCR, or random insertion of DNA-based transposable elements. Okay, so to do targeted mutagenesis in mice, one of the way we can do targeted mutagenesis is by homologous recombination. If we use a disrupted copy of the target gene, that means we have changed the copy of the targeted gene that will not make the correct protein now, and we use that for homologous recombination. That can disrupt the gene, our target gene. Okay. So that's very similar to what we already think. This is not the new material. This is from the previous test when we were talking about homologous recombination, and also we talked about CRISPR, how CRISPR uses this homologous recombination. Next question, next example question is how do SNRPs catalyze the removal of an intron from hnRNA, heterogeneous RNA. You remember SNRPs. These SNRPs are these RNA protein complexes. There's U1, U2, U4, U5, U6. These all bind together in order to splice the introns out of exons for RNA splicing. How do they do that? Let's look at the answers. They release the 5' end of the intron and use exonuclease activity to remove it before joining the exons. The SNRPs do cut the 5' end of intron, but they do not use exonuclease activity to remove it before joining the exon, right? Because what it does is intron, this is intron, this is the binding of this SNRP at this branch site will present this A for the lariat formation. This A will make a cut at this site here at the 5' end of intron, and there is now a hydroxyl group. That hydroxyl group will usually bind to the U6, but when this is not bound, when it's the time to make this cut here, that 3' hydroxyl group will make a cut here at the second site of the 3' end of intron, bringing the intron completely out. So this is the lariat formation here. This A, this presentation of A, or this A that comes out of the structure, kind of hangs out. This makes a cut here at the first at this junction, exon-intron junction. This 3' hydroxyl end now at the 5' of this intron will then make this cut here, and then this will intron will be spliced out. Okay, so no, there is no exonuclease activity here, so this is incorrect. The release of the 3' end of intron, so this one here, use exonuclease activity to remove it before joining the exons. That is also not correct, because there is no exonuclease activity here. This 3' hydroxyl group will make a cut here. Okay. The catalyzed sequential transesterification reaction to form a lariat and join the introns. This is actually the correct answer because this is a sequential transesterification reaction when this A here at the branch site will make a cut here at the 5' end of the intron, and then 5' end of the intron or 3' end of the axon, eventually make a cut at this 3' end, between 3' end of intron, 5' end of second axon. They form structure with recombinase-like activities that recognize splicing signals as recombination sites. Okay, so just to give you a little bit more detail of this transesterification reaction is the spliceosomal splicing, or this splicing of these introns. This involves a two-step biochemical process. Both of these steps involve transesterification reaction. What this mean is this transesterification is just two sequential reactions. First, this 2' hydroxyl of a specific branch point nucleotide. So here, this A within the intron, this will perform a nucleophilic attack at the 5' splice site in the intron exon boundary, and it will form the lariat intermediate or lariat formation. Second, this 3' hydroxyl group of the released 5' axon will then perform an electrophilic attack here. This electrophilic attack will lead to removal of this intron from the splice site, and these exons will then be joined. Okay. Let's talk about a little different question now. So if you have to transcribe and translate a DNA sequence, what will be the sequence of this transcribed and translated molecule? We haven't talked about translation yet, but we will be talking about before the test. So I just wanted to bring this up a little bit. And you already know the concept of codons. You know the codons code for amino acid. There's three nucleotides make a codon, and these code for a specific amino acid. So we have already done that. So here. So since this is so one thing to keep in mind, this is 5' to 3', right? So the mRNA will use 3' to 5' sequence as a template, and it will make exactly the same. The mRNA will be exactly the same sequence as the 5' to 3' template of sequence of DNA. So this is exactly the same sequence, except for instead of T there is U, because RNA molecules have U instead of thymine. They have uracil. The protein sequence for this will be MAGY. So methionine, adenine, glycine. So here, but why 4? So this should be 1, 2, 3, 4, 5, and 6 amino acid. But if you look closely here, there's a UAA at 5th position, and this 5th position UAA will code for a stop codon. So since it's a stop, this will not be translated, and since this will not be translated, nothing after the stop codon will be translated. So this will only have 4 amino acids for this sequence. Okay. So again, talking about transcription, which is the correct for termination of transcription in eukaryotes. So remember, we talked about termination of transcription is different, dependent or row independent in prokaryotes. In eukaryotes, because the mRNA needs to be post-transcriptionally modified, there are different ways of termination, or different set of termination mechanism in eukaryotes. Termination. So let's look at the answers, choices here. Termination after polyadenylation. That means termination will happen after the about 200 amino acid, 200 adenine nucleotides have been added to the end of mRNA molecule. Terminates during polyadenylation. So while polyadenylation is happening, it terminates. Termination prior or during or after polyadenylation, well, anytime, pretty much. termination prior to polyadenylation, or it will terminate before polyadenylation starts? The answer is after polyadenylation, because the 5' cap is added as soon as RNA comes out. Polyadenylation happens before the transcript will completely dissociate itself from the RNA polymerase. So that means that the polyadenylation happens before termination of transcription. And then the splicing happens after termination. What is the activity of integrase for a non-LTR retrotransposon? Now you remember we have 3 different kind of transposable elements, the DNA transposable elements, and 2 different type of RNA or non-viral like retrotransposons or non-LTR. What is the activity of integrase for a non-LTR retrotransposon? So remember, the mechanism of LTR and non-LTR retrotransposons is quite different, because in case of LTR cytoplasm, and then it comes back with the proteins into the nucleus, right? So what is the activity of integrase? It's bind to its cognate cDNA and bringing it back into the nucleus, binding to its cognate mRNA and bringing from the nucleus to the cytoplasm, binding to its cognate cDNA and bringing from the nucleus to the cytoplasm, binding to its cognate mRNA and bringing it back into the nucleus, binding to its cognate mRNA and initiating reverse transcriptase activity. Another difference between LTR and non-LTR retrotransposons is in LTR retrotransposons, mRNA is converted into cDNA. It's a second step, and then the second strand of cDNA will be formed before it attacks the new DNA sequence where it's going to insert itself. Whereas in non-LTR retrotransposons, the cDNA, the mRNA is actually goes and attacks